So today what we're going to talk about is some Biogas 101, basically what is biogas, how is it produced, and then we're going to go into just a brief history of the project here at Kettering, and then we're going to touch on some ongoing analysis and past projects, but really what I wanted to focus on today is some current projects that are happening in the lab that are funded and not funded. So, and then after we talk about these projects, then I'm going to talk about, hand it over to Steve Nartker, who is assistant professor in chemical engineering, and he is going to talk about membrane separation in a continuously monitored tank reactor that we've made in the lab. So first of all, what is anaerobic digestion? And we lovingly refer to this slide as happy peanuts. So on the left is aerobic digestion. What this is commonly referred to as is composting. So if you look over at Chevy in the Hole, that where all those leaves are, that's composting. That's where you break down organic material in the presence of oxygen. And what happens in composting is you lose a lot of that energy in those chemical bonds as heat. So when you see those piles steaming, when you look out your office window, MEs and chemistry people, that's the heat that's being generated from the breakdown of the organic material in the presence of oxygen. But what we focus on is anaerobic digestion. And anaerobic digestion is where you break down organic material in the absence of oxygen. So as we take in oxygen and breathe out carbon dioxide, these bacteria, or happy peanuts, aren't able to take in oxygen. There's no oxygen in their environment. So instead of giving off carbon dioxide, they give off methane. And instead of giving off that energy as heat, they put that energy into forming methane. So all throughout the talk, if you don't understand anything I'm talking about, just raise your hand. I'll be happy to answer any questions. And they lose very little energy as heat. So how do these happy peanuts fit into the research and what's happening at the full-scale plant? So these bacteria all reside in the anaerobic digester. There's 30,000 different types of bacteria that are in this oxygen-free environment. What happens is substrates are fed to this digester. The digester then has this bacteria. The bacteria eat the organic substrates. It's heated to about 37 degrees C. It's just like a cow's stomach. So this digester is basically the contents of a cow's stomach in a metal canister here. It's continuously stirred, and what's produced is a mixture of methane and carbon dioxide. This methane and carbon dioxide then can be used for energy generation. It can be scrubbed or cleaned up to purify the methane, which Dr. Nartker is going to talk about, or it can be used directly in CHP, or combined heat and power, to generate electricity. Another product of anaerobic digestion is a biofertilizer. So whatever you put in on this side, the organic substrates, like the food that you're eating, comes out on this side minus some carbon. Okay? So whatever you put in, you get out on the other side, but you take away the carbon. And you change the way that nitrogen is in the system. So you take nitrogen away from the organic molecules and just produce ammonia. So this reactor that's in the center here, there's different ways to operate these reactors. So there's three different, what we call processes that you can use to operate these reactors. The first one is typically what's occurring across the United States right now. And that is where you take manure or human waste and you feed it to the microbes, and then you can produce biogas. It has a less valuable fertilizer, and this is a pretty simple, straightforward process. What happens is that this type of anaerobic digestion is used to reduce solids content. So you can reduce the amount of solid that you have to treat and turn that carbon into biogas. So you're turning it into energy. The reason it's a less valuable fertilizer is because you're dealing with human waste. So when you deal with human waste, you have to worry about EPA 503B regs, which means where you can apply this human waste. You can't apply it to food crops we're going to eat. You can apply some to trees or greenhouses. However, there's a lot of monitoring that occurs when you're dealing with human waste and applying it back to the land as fertilizer. There's another process that occurs, and that is where you can take the waste, the cow manure or the human waste. And yes, we're talking about human waste. Okay? So just to get a giggle out of everybody while we're here. Get poop out of the way. Okay, poop's out of the way. Now we can start getting to the science. So here we have human waste or manure, but in addition we add some kind of other organic substrate, and we call these high-strength organic substrates. What this means is it's food that hasn't been digested yet. All of these other substrates is food that has been digested. So you can add this to the reactor, and you get more biogas because there's more energy in the non-digested food. It's a less valuable fertilizer, again, because you still have human waste present. But it's a more complex process because you have to deal with putting this effluent back into the plants. You have to look at ammonia loads at wastewater treatment plants and the regulations at wastewater treatment plants. The third process, and what we focus most on the lab, is operating anaerobic digesters without the need for any human waste or manure. And this produces the most biogas. It's a very valuable fertilizer. You don't have to worry about pathogens that are in there, human pathogens or human viruses. And this is a complex process. I would say most of the biogas plants operating in the United States are operating here. Most of the biogas plants operating in Europe are here. So they're operating plants that don't need manure and can use all high energy substrates. So this produces the most biogas, a valuable fertilizer. However, you have to monitor the process to make sure that the living organisms, those microbes, are happy at all times. So instead of, in this process where you can replace the microorganisms, here what we're doing is we're keeping the microorganisms that we have very happy. And the way we do that is with supplementation of trace nutrients. Okay, so we'll talk a little bit about that later on in the talk.
What's the source of that last one? Like you have waste as the first one. Is it just discarded or leftover food?
Yeah, so there's 27 million tons of food waste that goes into landfills every year in the United States. So this food waste that you're going to put in the garbage can right now, it can be fed to the digester to make biogas. And in fact, in the UK next year, they're banning all food waste to the landfills. And it's already been banned in Germany, Sweden, Austria, and the Netherlands. So it can be any kind of waste. I also have a picture of crops. Some countries are growing energy crops to put into the digester because they've used all their waste to generate energy, and they need some other source of energy in here. But most of the time, especially in the United States, we're just focusing on waste products. So what are these organic substrates or waste products? So all organic substrates are made of carbohydrates, proteins, and fats, macromolecules. And some of you students I've had in human biology should all know these macromolecules. And as you increase in complexity, you also increase in the amount of biogas that's produced. So here's manure. And for 1 ton total solids of manure, you can heat a home in Michigan for 23 days and how much biogas is produced. I wanted to put this in terms that you can relate to. Carbohydrates produces 15,000 feet cubed of methane, and it can heat a home for 60 days. Proteins, 71 days, and fats can heat your home for 134 days through anaerobic digestion and the production of methane. Just to give you an overview of what kind of substrates you can put in and what kind of energy values you can get out. And that's just from 1 ton of total solids of each one of these wastes. So here comes the biochemistry of what's happening there. So you knew you were going to get some biochemistry today. So what happens is all of these microbes work together. So there's 30,000 different microbes, and they all work together. And they process the substrates, the carbohydrates, fats, and proteins, in 4 steps. The first step, just like you're eating your sandwiches, the first step is to take that carbohydrate from the bun and break it down into sugars. And this all happens outside of the bacteria, so in the liquid, just like it happens in your intestines to break them down. And then those monomers are absorbed into the bacteria. That forms organic acids, alcohols, and aldehydes. There's also, it can form hydrogen and carbon dioxide, which can go straight to methanogenic bacteria and produce methane. These organic acids, alcohols, and aldehydes then make acetate. This acetate then can be broken down into methane and CO2. But our organic substrates contain carbon, hydrogen, oxygen, which we've accounted for with methane and CO2, but they also contain nitrogen and sulfur. So those have to come out on the other end. So the sulfur comes out through the sulfate-reducing bacteria and produces hydrogen sulfide. Hydrogen sulfide is a nasty, corrosive gas. Okay, so we have to meet, some of you are shaking your heads with experience. So we have to mediate that in the digester. And the nitrogen comes out as ammonia. So it gets cleaved off of the amino acids and gets released as ammonia in the digestate, or in the liquid part. The boxed chemicals are released in the biogas. So now that we've had Biogas 101, I thought I would start you on the history of where this project is. And more importantly, I want to spend most of my time talking about where we're at now. So remember the King of Sweden visited Kettering with Jennifer Granholm, and we formed the Center of Energy Excellence, which is a partnership between Swedish Biogas, Kettering University, and the City of Flint. It was funded through the DOE and MEDC. Well, we've continued that collaboration past the funding, and we've started the collaboration with Linköping University in Sweden, thanks to the Oswald Fellowship, I was able to travel over there, and we submitted a proposal to the Swedish Energy Agency to create the Biogas Research Center at Linköping University. This is a $21 million project that is solidifies the collaboration between Linköping and Kettering University. And we should hear back any day from this collaboration, but Sweden's about 90% sure that it'll get funded. So we'll see, keep your fingers crossed to see what happens. But we wanted to continue this partnership and expand the partnership to Linköping University. As part of this partnership, as most of you know, Swedish Biogas built the biogas plant out at the Flint wastewater treatment site. It's a 1 million gallon digester. And this is a picture standing on top of the digester looking out at the digestate storage tank. It processes 100% of this Flint sludge. So anything that you go to the bathroom here gets put in this digester. There's 90,000 cubic feet of biogas per day that'll heat one home for 250 days here in Michigan. It reduced the solids amount, so they could close down 2 of the incinerators that they have. And the project saves the city about $250,000 a year.
So where is that gas going?
Right now, it's being used to not only run the digester, but then they're looking into ways to use it to generate electricity. But more importantly, I wanted to talk about what's happening over at the Innovation Center. So since the collaboration between Kettering and Swedish Biogas went well, we decided to collaboratively open the lab in the Innovation Center. We call it the North American Advanced Biogas Laboratory, or the NAB Lab. And I'm going to talk about some of the projects that are ongoing in the lab now. But first, before the students are laughing, before I talk about the projects, I really want to talk about the people that are doing the work. Okay, these are our undergraduate students here at Kettering. So I'll just briefly go through. Mandy Anderson graduated 2 years ago, a year and a half ago, and she's at Emory University. Steve Needham is at JHP Pharmaceuticals. Ben Wagner is here, and he's an electrical engineer for Swedish Biogas. Silky Color is a undergraduate that did an exchange program from Rutgers University and now is coming back to do her thesis in the NAB lab. Tiffany Snow, you can raise your hand. Yeah. Tiffany Snow is a senior here at Kettering, and she's doing her thesis with me, looking at food waste from hospitals. Olivia Hayden is here, and she's a senior at Kettering, and she's also the lab technician from Swedish Biogas. So she was hired by Swedish Biogas. And I'll talk about the stuff that she does in the future. And then we have Andrea Matuska, who graduated. She was a biochemistry, chemical engineer double major. She's at University of Miami in Florida, doing graduate studies. And I also have Katie Hoff here that she started an algae project, and I'm glad to see her here today. And we've actually been able to continue that project, so I'm glad you're here to hear about that. So here is the some of the capabilities we can do in the lab and what we've done in the past. So we can determine the biological methane potential of any organic substrate. So what that means is, if you give me this cookie, how much methane can I make from it? Okay. So, okay, I knew some joke like that was going to come out during the talk. I have to tell you, my 8-year-old thinks that it's the coolest thing that his mom works with poop every day. So, methane we can make in the lab, okay? So this is different substrates, and we've looked at over 40 substrates, probably more in the lab. Some of them we've looked at is seed corn and fungicide-treated seed corn. Will the fungicide kill the reactor, kill the bacteria? Different corn grinds. Do you have to spend the energy to grind corn down in order to put it in an anaerobic digester? Or can you just leave it as a coarse grind? We looked at DAF, which is dissolved air flotation, which is basically all the fat that floats to the top of water that you scrape off. That's not our favorite substrate to work with. And then you can see that the methane production is tallied over time. And some substrates produce a lot of biogas, like this blue one, but takes longer amount of time. And some substrates, such as yogurt waste, produces less biogas but produces it more quickly. So we can look at the kinetics of this BMP, look at the rate of biogas production, and then look at how much biogas they're going to produce. We can also look at these substrates in small-scale tank reactors, and we name each one of them. This is Caroline. And we operate these, we feed them daily, we look at biogas production, and we can test different recipes of what kind of food we're going to put into the reactors. We've looked at primary sludge. We've looked at the second case of primary sludge with different wastes. And then we've also looked at the mono substrate, where you just put in one substrate to the reactor, and looked at the biogas production. So this is really where the focus of the lab is looking at one substrate into the reactor and being able to maximize the biogas production from that. Every week we test those for pH alkalinity. I'm using the royal we. Olivia tests this for pH, or the other students working in the lab. pH alkalinity degradation, BFA, sulfate, TKN, ammonia, and phosphate, just so we can get an idea of the health of the reactor. The first thing that goes is you're going to get high volatile fatty acid production. If you get high volatile fatty acid, it's just like having an upset stomach. It's going to throw up. So these reactors have thrown up over the top. That's not something you want to happen in a full-scale plant. That's why we do this. That's why we do this at small scale and test out our process additives at small scale, or test out our recipes at small scale. That way, you're not having these problems at the full-scale plant. And we also look at gas production, methane and carbon dioxide percentage, and then we also look at trace components in the biogas. So we can operate 2 reactors within a 3.2% relative standard deviation. Because these are microbial reactors, we have to make sure that we can operate them and feed them the same so we can compare the data between the 2. And that's pretty good match between 2 reactors. We can also operate within 4.7% relative standard deviation to the full-scale plant. So the full-scale plant and these small reactors, we want to make sure that what we're doing in the small reactor mimics the full-scale plant. So this data can be extrapolated. Okay, so why the heck do I have Diet Coke, shiny hair, and deodorant on my next slide? Well, we're looking at trace components in biogas. Okay, so we've made the reactors We looked at the methane potential. Now we're going to look at trace components that are coming out in this biogas. What's in there besides methane and carbon dioxide? And one of the big problems, especially working with wastewater treatment, is everything that you put on your hair to make it shiny. Okay, so all of these shiny ingredients are siloxanes. These siloxanes are found in deodorants to make them smooth. They make your hair shiny, right, Jamie? and they also, what happens is that these siloxanes get washed off in your wastewater. So when you take a shower, they go down the drain, it goes to the wastewater treatment plant. When it goes to the wastewater treatment plant and that sludge goes into the digester, what happens is that these siloxanes, which are up here, volatilize. They get into the biogas. Once you burn these, they turn into this, sand. So once you burn siloxanes, they turn into sand. And when they turn into sand and you put them in an engine, most of you know what's going to happen, right? So the engine's going to break down, you're not going to be able to use this biogas, and you've lost thousands of dollars on your Jenbacher engine. So what happens is that as a thesis project, Olivia Hayden has determined the quantification and identification of siloxanes in biogas. So she's able to take the biogas off the plant, off of landfills, and is able to determine the presence of them, and also to quantify both the cyclical and linear siloxanes found in the biogas, which saves biogas producers lots of money to look at siloxane remediation. So that was her thesis project that she's writing up now. So now I'm going to spend the last 10 minutes, and I thought I'd have more time to go over the currently funded projects, or projects that are ongoing in the lab right now. The first one is looking at food waste. And this food waste is from Genesis Regional Medical Center. It's funded by the Michigan Biomass Energy Program. And the reason we chose hospital food waste is that it's point separated. Meaning there's 4 ladies at this particular hospital that scrape all the food off of the plates from the patients. And they can separate out the silverware or the plastic or the paper. So it's a waste product. Typically it's landfilled or composted. Most hospitals are still landfilling. And the published report just stated yesterday that there's 27 million tons of food waste a year produced in the United States. So that's a great opportunity to be able to put this in a digester and to make biogas from. The approach is that we're doing a waste and chemical analysis. We're doing the BMP to determine how much methane is produced. We're doing the mono-substrate small-scale reactor. I'm partnering with Swedish Biogas to do the economic analysis. Is it economically feasible to do a full-scale plant with food waste? And also outreach to hospitals and universities about the potential of their food waste to generate biogas. The big challenge with this research is that unlike other digesters where we feed the same recipe every day, the food waste changes every day. So these bacteria are going to see different food every day. So the goal is to monitor this reactor to make sure that it can handle this constantly changing substrate. So here's some preliminary results. Here's a picture of me scraping food waste at Genesis Regional Medical Center. Tiffany Snow and I, you can see the students' names down in the right-hand corner. Tiffany Snow has also scraped the food plates with me, and she is working on me with this project, and we looked at a week of food waste, and we calculated that they are throwing away 3,869 pounds of food waste a week. We've started a small-scale reactor, and we're doing the biological methane potential test right now on different food wastes. So that's a great opportunity to use food waste, but also to look at it in the mono-substrate reactors. We've also received a grant from the Michigan Soybean Promotion Committee, and since soybeans are the second largest crop in Michigan, and it's unique because it's an oilseed crop. A lot of times oilseed crops aren't used in anaerobic digesters. And since there's no true soybean or large soybean cultivation in Europe, where most of the anaerobic digestion or biogas research occurs, we wanted to look and see how soybeans react in an anaerobic digester to produce biogas. And again, we're going to run a mono-substrate digester and do a fertilizer analysis. Because right now, farmers, what they do is they undertill the soybean residue, and they want to make sure that taking the soybean crop, not the seed, but the leftover plant off the field is going to be okay, and that they have this fertilizer to add back to the soybeans or to the field. And again, Swedish Biogas will do economic analysis with this project. And the challenges, again, are the undertilling versus the fertilizer. Another project that we're looking at is a biorefinery concept. So I'm partnering with Sar Krovchak from University of Kentucky, and she has a photobioreactor system operating off of algae and being fed CO2 from a coal-fired power plant. So she has this algae growing with CO2, and this is a self-flocculating algae, meaning that it's going to settle out by itself. You don't have to dry it or spend the energy to dry it. So we can decant off the liquid and then take the algae and put it into a digester. And then we made it an integrated system, and we submitted a grant to the National Science Foundation for Energy for Sustainability and hoping to hear back from that soon. But she has funding from the Kentucky Energy Agency, and we're looking at the algae in the anaerobic digestion. So here's a picture of our photobioreactor system. Here is the BMP, so how the algae reacts in the anaerobic digestion or biogas system. So this is the accumulated amount of algae. There's no long-term studies on algae and anaerobic digestion, and we're looking to construct the anaerobic digestion or biogas facility at the University of Kentucky. So we're partnering to do this. The challenges with this project is the biogas production is only giving about 20% degradation, so we're only eating about 20% of the biomass, because algae, it is naturally bacterial resistant. It doesn't want bacteria to eat it. So we have to find some way of letting these algae let the bacteria eat itself. So, and another problem is with algae, a lot of the proteins on the outside of the algae have a lot of ammonia or a lot of nitrogen in them, so you get ammonia accumulation in the digester. So those are two challenges that we're trying to overcome to install this full-scale plant at the University of Kentucky. I'm doing pretty good, 4 minutes. So some other projects that we want to start up in the future or have worked on currently is I've worked with Daniela, I can't say her name, for mathematics to look at the statistical analysis of volatile fatty acids. And she actually found a problem with our method, and we were able to correct it. So now we're going back to refine the method for looking at volatile fatty acids. We're looking at Tiffany, I worked with Tiffany to look at protease inhibition to stimulate hydrolysis, meaning how can we break down that substrate quicker so that we don't have to leave it in the digester as long. This summer when Silky comes back, we're going to look at kinetics of the reaction, looking at how to determine the kinetics of different substrates in the biological methane potential test. And again, we're going to look at methanogenesis additives. Methanogenesis bacteria use some really cool inorganic molecules. So they use molybdenum, cobalt, iron, nickel, selenium, tungsten. So these are limiting in the anaerobic digester, especially when it's mono substrates. So we want to look and see if maybe these substrates, these inorganic materials are needed to stimulate biogas production. Okay, so I think I've talked a lot. Is there any questions before we go on to, yeah, Jonathan?
Yes, you've been mentioning that ammonia is a problem in your process. Have you considered actually capturing that? Because that actually has some value to it.
Right, so the ammonia that comes out in the biofertilizer, it's not a problem until it reaches inhibitory levels. So when it reaches inhibitory levels, then that's when it causes inhibition. Right now, it's not a problem, it's actually a benefit in the fertilizer. So we want to get as much ammonia as we can, but not inhibit the process. So the idea is that we want the ammonia in there because it is a value to the fertilizer and it provides buffering capabilities. So one thing I didn't say was these reactors, it's a biological system, they buffer themselves. They keep them at pH 7.6. So the ammonia participates in the buffering capability. So if we remove the ammonia, then it won't be able to buffer itself, and we'd have to supplement with calcium carbonate or something to buffer itself. Yeah.
Environmental advantage, efficiency, how about the efficiency will go into this process and what is getting from?
Right. So we've looked at mass balance and we've looked at the energy content. And if it wasn't an efficient process, Swedish Biogas wouldn't be a business. So you make more energy from the biogas than it takes to run the operation. So you're making plenty of biogas, especially when you're adding the substrates to the instead of just the waste, the human waste and the manure. When you add the high strength organic substrates, you are producing a lot more biogas than you need to operate the digester. And we've done, go ahead.
What is the efficiency?
Like the parasitic load on the plant. So at the, do you want to help me with this?
Which plant I think normally you use like 10%
Yeah. So
of the energy production in the biogas. If you have a co-digestion like a large industrial facility.
So let's say in Sweden, at the plants that are just doing pure co-digestion without manure, the amount of biogas they have to use to run the plant is about 10% of the biogas production. When you're using wastewater, of course, it's a little more because you're not getting that high energy value content. Yeah, Stacey.
Did you ever approach Sodexo?
It's funny because we said someone's going to mention that. So, as part of the Michigan Biomass Energy Grant, we said that we're going to do outreach to universities to show them the potential of biogas production from food waste. So yes, we will at the end of this grant once we get all the data. Yes.
Are you at a point where garbage that's collected is separated? Are there plants or something that separates them already for you?
Most of the time, and in looking at the models for anaerobic digestion, those that are advanced, like Sweden and Germany, it's separated at the household and then collected separately. We're first focusing here on our research is looking at waste that is separated by one facility, so like a cafeteria or a concentration of people. That way your waste is very clean. We've looked at food waste from households, and the problem with that is that there's bags and silverware and other things that are in there, where if it's the employee's job to separate the food at the facility, then there'll be a much cleaner resource. And there's one other thing I wanted to do too is I didn't thank Andre and for his help with the siloxane project with Olivia, with his help with her thesis. I want to make sure I got that in there. Anything else? Okay, I'm going to turn the floor over to you.
All right, thank you.
And I'm going to turn.
All right, first of all, I need to thank Dr. Aurandt. Without her, I wouldn't be doing the research that I'm doing now, so she kind of took me under her wing a year and a half ago when it all started. And I'm going to get it started by showing a video. This is Ben Wagner.
I built my own model scale reactor for my thesis project and kind of just tried to automate the data logging of everything so we could really get a better picture of what's going on a day-to-day basis. Currently they just take a reading off the gas counters once a day, so it's very labor intensive as far as having someone in the lab all the time to monitor them, and you really only get a snapshot of once a day how much is this thing producing, where my reactor now you can see the methane production in real time and see how, like, because normally after you feed it will spike the methane production and then the gas production will kind of fall off until you feed again the next day. So I think really it'll just give us a better picture of everything that's going on.
What Ben's talking about is, sometimes it takes the combination of biochemists, electrical engineers, chemical engineers, pretty much everybody to get the whole project going the way you want to go. So what Ben developed is this apparatus here, where you can record the mass. So the reactor, it's a 5-liter glass reactor, it's just sitting on a balance. It's continually recording the mass. You've got a motor that's running a stir paddle. You have a tachometer that's hooked up to that, so you're always know your stirring rate. And then we have 2 gas meters that come out. So the biogas that's produced from the anaerobic digestion, we're looking at the amount of methane and the amount of CO2 that's being produced. It all goes into a data acquisition system and gets logged onto a computer. So tying into Dr. Aurandt's work, we want to know if we change the diet of the reactor, how does that affect methane production, what's the time lag, and that sort of thing. So where we want to go with this is we know that methane is being produced. How do we get some useful work out of the methane or the gas that's being produced? So again, it's a combination. It's a multidisciplinary approach where we take people from multiple backgrounds: chemistry, biochemistry, chemical engineering. The names have been mentioned before: Andrea Matuska, Olivia Hayden, Silke Keller. But these are the people that are feeding the reactors daily, doing all of the monitoring and analyze work. We also use these reactors for examples in chemical engineering courses like mass energy balance. So it's a real world example of what's going on. This is the user interface that Ben developed. So instead of going through the process like he said, and just having one day where you say, okay, this is the gas production, at X amount of hours, this is a continuous monitoring program. So at any point in time, you can see where your values are, and if you export it into Excel, you can make charts and that sort of thing. All right, so the difference between biogas and natural gas is, unfortunately, when it goes to the anaerobic digestion process, you don't get natural gas out. As Dr. Aurandt mentioned, there's some impurities in there. Okay, some of those impurities are siloxanes, hydrogen sulfide, that sort of thing. Okay, we know natural gas, so that's what we use to heat our homes. If we compress it, we can use it as a transport fuel. We can also do the same with biogas. The problem is we just need to go through a little bit of refinement. Okay, so the refinement really is kind of site specific. If you're comfortable with a low energy value of biogas that has 60% methane by volume, for example, has a calorific value of 600 BTUs per cubic foot, that's useful gas if you want to run a combined heat and power cycle. Okay, but it's not useful if you want to run internal combustion in an automobile. So where you stop on this upgrading process is dependent on what the end application is. Okay, so at the full-scale plant, what they do is a combined heat and power cycle. So you can actually have a gas that has a lower methane content. You put it into the CHP, which is basically an engine hooked up to a generator. The engine runs on the methane, spins the crankshaft, which goes inside the generator and produces electricity. Okay, so at the point now where there's an excess. Okay, so a plant can be self-sufficient and produce actually more electricity by burning the biogas. All right, so what can we do with the excess biogas that's not required to run the plant? We can upgrade it again. The goal here, the ultimate end goal would be to upgrade the biogas so that you have something that can replace natural gas for transport and for stationary sources. Okay, there's lots of different ways you can upgrade biogas, and I'm going to compare and contrast several of these. Many of these are proven in the industry to work and be effective. Okay, it's usually based on costs and availability of materials, and it's pretty much a localized concern of what you want to do. The typical ones are chemical absorption of hydrogen sulfide and CO2 into an amine solution. This is like when we do our absorption process in our chemical engineering labs. We have high pressure water scrubbing, pressure swing absorption, cryogenics, and finally membranes. And I've chosen to focus on membranes for a couple reasons, which will become apparent in the next couple slides. Okay, so this probably looks like an eye test to most, but basically what this is saying, it's a comparison of those 5 separation technologies that I showed before, from cryogenics being the most expensive to run based on investment costs and day-to-day costs, down to membrane separations, which are actually the cheapest, and the same thing on a price per cubic foot basis. Okay, so the biogas that's produced after going through a membrane process is actually the cheapest biogas that you can get. Of course, anything in engineering, there's got to be a drawback, right? It can't be all advantages. The drawback is the performance. The performance of membrane separations right now is lagging behind the other separation technologies. All right, so that's where we at Kettering and our collaborations that we've developed, we see the need is to increase the performance of the membrane separations. So we're working with couple of companies, and we're also working in our lab to make the membrane separation process more efficient, or as efficient as possible. This started with Andrea Matuska, who was a chemical engineering biochem dual major. She set up the first membrane separation device over in the NAB lab. Okay, and what we did here was just take a pressurized gas. It's a synthetic biogas mixture, so it's 65% methane, 35% CO2. You regulate the pressure and you regulate the residence time of the gas in the membrane and collect samples. Okay, so this is a more detailed picture. We just have more flow meters and stuff like that. But basically it goes from Andrea's project of setting up the membrane separations, okay, down to Olivia's project where it goes into a gas bag, and Olivia takes the gas bag to the GC and tells us what's in there. Okay? So what's actually going on inside the membrane is that it's filled, well, you can have several different types of membranes. The membranes that we chose to use are a hollow fiber membrane. So the raw biogas comes into the end at a high pressure. It flows through a hollow fiber that does two things. It limits the, it acts like a mechanical filter, filtering out larger molecules from smaller molecules, and also as a permeation medium. So the CO2 dissolves more easily in the polymer than the methane does. So that allows CO2 to come out the permeate side and the methane to be retained in the high pressure stream. Okay, this is kind of an exploded view of what these hollow fiber membranes look like. As I mentioned before, right now the performance is kind of a little bit behind the curve in terms of the other technologies that are out there. So what people do is they install them as modules in either series or parallel to get the methane content that you want in your final application. All right, so like I mentioned, we did some performance analysis on commercially available membranes, and all we did was we varied the pressure control. But to tell you, you can also vary the temperature and the sequencing of these membranes. And we started with a synthetic biogas. We set the regulator pressure at 100 psi. That's where we maxed out right now. These membranes can, or the pressure housing can go up to 350 psi, but we're just not there yet. And then we collect and analyze the sample bags. Okay, as I mentioned, this was Olivia's work. I'm kind of fast forwarding a little bit now. But yeah, the future is going to be to do more pressure runs and develop a continuous gas monitoring function. So similar to the work that Ben did on the small scale little anaerobic digester, instead of just piping the membrane effluents into gas bags and then doing it in a batch, take it to the GC and analyze that, we want to develop something so we can continuously monitor the concentration of the gases that are going through the membrane. But today, there's good news. At 100 psi, we can get a methane concentration of 94%, which is pretty good. It's proven repeatedly in lab, and as low as a 20% methane slip. The methane slip means the permeate side, we're also getting methane through, but we'll address that in the future by sequencing membranes and that sort of thing. It looks like I'm being done. So the opportunity is to help develop new technologies, be on the forefront of where we are in terms of a low-cost operation, develop new technologies, new materials so that we can get the performance up there where it needs to be and fit into the whole energy cycle. We need to give some acknowledgements now? Okay. I'd like to thank Dr. Melton, Dr. Seeley, Provost Simpson, and Provost Harris for all their support. And
So we'd like to thank Daniela too for her help with the project with the statistics. And SBI, of course. SBI, I'd like to thank Anaerobic process engineer. The reason this partnership works is because her and I can manage the lab together and balance everything, keep everyone in line. And Tom Guy is the CEO, who's been really open to collaboration with Kettering University and is more than happy to host anyone over at the lab. And now that Olivia and Ben are employees of Swedish Biogas, thank you to you for allowing us to work in this space. And also our collaborators, Dr. Krocek from University of Kentucky and Dr. Seeley from Oakland University. Yes, Dr. Seeley's husband. He's the one who determined the method for separating methane and carbon dioxide in the lab that you saw Steve produce. And hopefully a potential future collaborator Dr. Albert's here from Oregon State University. We would like to end on this slide for questions, because this is really SBI's vision and Kettering's our vision of what we call beyond biogas, how biogas fits into the environment, industrial symbiosis, how we can use biogas to grow algae from the CO2 to provide methane for vehicle fuel, to use the substrates as waste products, to use the biosolids for fertilizer to grow the substrates that are going to be fed into the digester. So this is our vision. This is what's already happening in the Twin Cities of Linköping in Sweden. I know I didn't say this right.
Linköping and Norrköping.
In Norrköping, sorry, Norrköping. So this is what's happening already in Sweden, where one industry feeds another industry. And the model there is waste is just a resource misplaced. They don't have landfills. They don't put things in the ground after they're done using them. So what they do is they have a combined heat and power plant that incinerates their waste and uses that for district heating, so they heat their homes with the waste that they incinerate. And in fact, today, there's an article in the New York Times that said that New York is asking for proposals to use their waste to turn it into energy. So I think that the United States is going in that direction to use waste as energy. So with that soapbox speech.
I need to say three things before we open up to questions. And for those who can't be with the class, first of all, thank you very much. You really exemplify the research that's happening here in Flint, Michigan, for the Kettering community, but really for the larger world. And what you're doing is so important, so thank you. And